Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we started discussing immunology, in which yesterday we discussed about the properties and hallmarks of the immune system, the basic features that our immune system has, as well as we also learned about different types of immunity that we possess. The one with which we are born, the innate immunity, and the one which we develop over the time due to the exposure to different pathogens, the adaptive immunity. So we had kind of introductory lecture about the immune system, what is immunity and what are the types. So in today's lecture, we are going to learn about these cells from starting today, we are going to learn about the cells of immune system, right? So the cells which are the warriors, which are going to protect the host against the pathogenic attack. So in today's lecture, we are going to learn about hematopoietic stem cells and hematopoiesis, means the generation of blood in detail. So we will be learning about different, how the process of hematopoiesis goes on from the hematopoietic stem cells. So before starting this lecture, I would like to mention a few terms. I will explain them to you. I was going to explain them in stem cell biology, but I think it is important for you to understand those here because in this immunology series, I will be using those terms a lot. So let's start with the lecture. So a few terms that I would like to introduce you are the different categories of the stem cells. Okay, so based on the potency of differentiation of the stem cells, they are categorized into four different types: the totipotent cells, pluripotent cells, multipotent cells, and unipotent cells. So these cells are categorized on the basis of the ability of that particular stem cell to form different cell lineages. For example, the very first category is totipotent cells. Okay, so the totipotent cells are the ones which can give rise. So all of them are the stem cells. So stem cells has the ability to regenerate themselves, unlike the normal cells. So they can regenerate themselves, and this property is known as the renewable property. So they can renew themselves, so as well as they also can be differentiated to form multiple cell lineages. So that is what stem cells are, right? So unlike the normal cells which can divide until certain limit, which is known as Hayflick limit, stem cells, they can basically regenerate themselves and they can keep dividing to form different type of cells. For example, the zygote, which is a single cell which is formed after the fertilization of two haploid cells, sperm and the ovum. So once egg and sperm, they fuse together to form zygote, the single cell can give rise to full organism, right? The whole individual. And this individual have different types of cell, neurons, muscle cells, the immune cells. So all those different types of cells have different structure, function, different composition, right? So the ability to give rise to the whole organism, that ability is of the, that ability is possessed by this stem cell. And this type of stem cell which can give rise to whole organism is known as totipotent cells, means they have the whole potency. So single cell, it can form cardiac muscle cell, it can form the normal muscle cell, it can form the neuron cells. According to the conditions that are being provided to those cells, it can give rise to any type of cell. That cell, that stem cell, is known as totipotent cell. So the example for totipotent cell is the zygote itself. These are the cells which can give rise to whole organism. Okay, so they can give rise to embryo. So this zygote is going to form embryo in future. It gets developed into embryo after multiple cell divisions, and it also forms extra embryonic tissue, which is going to form placenta, right? So both embryo and extra embryonic tissues can be formed by totipotent cells. The next category of the stem cells is pluripotent cells. Okay. So the pluripotent cells, you could say that those cells can give rise to all cells that are the that are there, that exist in the individual, except the extra embryonic tissue. So except the placenta, pluripotent cell can give rise to any type of cells that could be found in the individual. So we can also say that pluripotent cells are the embryonic stem cells. Okay, or we could also say that those are the cells which can give rise to all cells except placenta, except the extra embryonic tissue, right? The next category of the stem cells is known as multipotent cells. Okay, these are also referred as adult stem cells because those stem cells can give rise to multiple lineages, but they cannot, they cannot form the whole organism. So they can differentiate into multiple cell types, but the lineages they can, differentiate into, that is limited. So they can, these are the cells that can differentiate into limited number of cell types. Okay, so that is why they are also known as adult stem cells. Okay, so unlike the embryonic stem cells, which can give rise to the whole organism, to the whole different types of, not the whole organism, sorry, to the different types of cells that can form, that can be found in an individual, except the extra embryonic tissue. Well, for the development of whole organism, you need extra embryonic tissue as well. So pluripotent cell, they cannot develop the whole organism, but they can develop all type of cells that can be found in that particular organism. On the contrary, adult stem cells, they can only differentiate into limited number of cell lineages. So they cannot really form the all type of cells that can be found inside the, inside an organism, in an organism. So multipotent cells, they are, more differentiated version of the pluripotent cell. We can also call that. And the last type is known as unipotent cells. Okay, so the most typical example for the multipotent cells is the hematopoietic stem cells that we are going to discuss today. So hematopoietic stem cells, this is the stem cell which is going to give rise to multiple lineages such as myeloid lineages, lymphoid lineages, which are going to form the cells of blood that are found in blood. So blood, which is a heterogeneous tissue, it has multiple cells such as WBCs, RBCs, platelets. So all of them, they are different cells. They have different characteristics. So they belong to different lineages. So all those different types of lineages, they can arise from hematopoietic stem cells. But this doesn't mean that hematopoietic stem cell can give rise to a muscle cell, right? So this is how multipotent stem cells, they are able to give rise to multiple type of cell lineages, but that number of the cell lineages which they can differentiate into is limited. Okay, so the example is hematopoietic stem cell, muscle stem cells, or okay, so those can give rise to only one type or more than one type of cell lineages, but not all of the cell lineages that are found in the adult individual. Okay, and on the contrary, unipotent stem cells. Oh well, by mistake I called muscle stem cells. They are not multipotent. The muscle stem cells, they are unipotent cells that I'm discussing now. So unipotent cells, unipotent stem cells are the ones which can differentiate into only one type of cells. For example, the muscle stem cells. So muscle stem cells can only form the muscle cell. Okay, they cannot give rise to cardiac cell, or they cannot give rise to neurons. They can only form muscles, but they are stem cells because they have the capacity to regenerate themselves again and again. So this type of cells are known as unipotent cells. So they can give rise to single cell type. Okay, so unipotent cell, they can renew themselves, or the daughter cells of unipotent stem cells, they can differentiate into single cell type. So only single cell lineage is going to be there as a result of the unipotent stem cell division. Okay, so the example is muscle stem cells. Okay, so the muscle cells, they are going to give rise to only muscle cells. So these are the different types of stem cells that are found in the that are found in an individual at different stages of the development. So the pluripotent stem cells, they are very important because they can be used for the formation of transgenic mice. So well, transgenic mice is those are the mice whose genome have been altered by introducing some foreign DNA. So I'll have a separate lecture about the how we can form or create those transgenic mice, and how from those transgenic mice we can form the knockout mice, knock-in mice, which are really important for conducting the immunological experiments. So we can create the conventional knockout mice or the conditional knockout mice. So we'll have a separate lecture. So for formation of those transgenic mice, we need the pluripotent cells. So those pluripotent cells are taken, and they in those cells we use them to the pluripotent cells that are extracted from the mice. They are taken from the blastocyst stage, the developmental stage blastocyst, and then they are used for formation of transgenic mice. So this is an important point. Please make a note of it because it will be helpful in the future lectures. So I hope those different terms, those different categories of the stem cells are clear to you all. So now we are going to discuss about hematopoietic stem cells, which are going to give rise to the all blood cells, different types of blood cells. In those blood cells, there exist the immune cells. Okay, so that is why it is important to know about hematopoietic stem cells. So let's learn about hematopoietic stem cells. So as discussed already, that hematopoietic stem cells, they are the multipotent stem cells, means they can give rise to multiple cell lineages. So those hematopoietic stem cells, when they differentiate and they mature into the blood cells, so when they get differentiated and mature into the blood cells, this whole phenomena, this whole process is known as hematopoiesis. So by the end of this lecture, you will know a lot of things about hematopoietic stem cells as well as hematopoiesis. So today we are going to discuss those two topics, the HSCs and the hematopoiesis. So the hematopoietic stem cells, they are found in blood marrow, bone marrow, sorry, bone marrow. Okay, so the number is very less in the bone marrow. So from the one cell, single cell in each 50,000 bone marrow cells is found. So like if you take a pool of the cells which has let's say 1 lakh or a 50,000 cells, 50,000 bone marrow cells. In those 50,000 bone marrow cells exist a single hematopoietic stem cell. So the strength of hematopoietic stem cell in the bone marrow is very, very less. So they are very rare, and that is why the isolation of hematopoietic stem cell, that was a really challenge for researchers in past. And due to the technical advancements such as the invention of flow cytometer, so using flow cytometry and using the formation of monoclonal antibodies, researchers, they were able to isolate those hematopoietic stem cells, because it is really, really difficult to isolate a single cell out of this whole pool of the blood cell. And again, whole pool of bone marrow cells, sorry. So this hematopoietic stem cells, they are rare, but their ability to differentiate, their ability to form the blood cells, is it's very enormous, it's very high. So a single, if you they have enormous proliferation. So if you infuse a few cells, less than 10 cells, they can recreate the whole blood pool that could be there in an individual. So this was proved by, so we can use a mice, okay, and let's say that this mice has been, it is exposed to the radiation, let's say 950 rats X-rays. So if we expose this mice to X-rays, the all of the hematopoietic stem cells that are there in the bone marrow, they are going to get killed. Okay, so this mice will die eventually due to lack of forming new blood cells. But if we infuse, let's say 10 raised to 4 to 10 raised to 5 bone marrow cells, this mice can be saved. So for escaping from the situation, from escaping this fatal situation, infusion of 10 raised to 4 to 10 raised to 5 bone marrow cells is fine. It's fine. So in those cells, in this large pool of bone marrow cells, there exist only a few, like less than 10 hematopoietic stem cells, and this particular number of hematopoietic stem cells, it's more than sufficient to create the whole pool of the blood cells. It can regenerate, it can proliferate enormously to create to have the whole blood cells formation formed again in the individual, and to save that individual from a fatal condition. So this is from this you can get the idea about the proliferative capacity of the hematopoietic stem cells, right? So when the individual is in its steady state, means when there is no infection or when there is no need of hematopoiesis, that is what we refer as homeostatic condition. So in the homeostatic condition, those hematopoietic stem cell either they renew, okay, or they form the progenitor cell. So they divide those hematopoietic stem cells. Okay, hematopoietic stem cells, they divide rarely, and that as they divide rarely, that is why the strength of those hematopoietic stem cells inside the bone marrow cells, it is very less. As I told you, one in each 50,000 bone marrow cells, it is really less, because they divide rarely. But once when they divide, so they divide rarely when the individual is at homeostatic conditions, means there's no need for there's no infection, so there's no need for hematopoiesis. So when the individual is in the homeostatic condition, the stem cells they divide rarely, but when they divide, those hematopoietic stem cells either they can renew, okay, those HSC they can either renew, okay, or they can form a more differentiated version, which is known as progenitor cell, which is which is committed to a certain lineage. It could be committed to myeloid lineage or lymphoid lineage. So progenitor cell is going to give rise to different cells, which are precursors of the blood cells. They are also stem cells, but they are they could be they are unipotent stem cells. So the progenitor cell, let's say it's a basophil progenitor cell, means it is going to give rise to only basophils, right? So the division of hematopoietic stem cells. So hematopoietic stem cell, when it divides and forms two daughter cells, either this daughter cell can renew itself, means it can sustain the capacity of regenerating itself, means it can still be a stem cell, or it can decide its commitment to certain lineage, so it can form a progenitor cell. Okay, so this decision is solely depending upon the activation of certain transcription regulators. I'll be discussing that as well, the GATA2, BMI1, Icaros, and NOSH1. So depending upon the activation of those transcription regulator, the daughter cells, they decide whether they are going to stay as hematopoietic stem cells, or they are going to get committed to certain cell lineage and get differentiated into a progenitor cell. Okay, but when the individual is in the in some stress condition, let's say it has exposed to infection, or it is undergoing chemotherapy, right? So in the chemotherapy, when the cells are being killed, there's demand of the hematopoiesis, right? Or even in the infection, the individual needs hematopoiesis. So whenever there's a condition of hematopoiesis, hematopoietic stem cells are going to show enormous proliferation, as I already told you. And this enormous proliferation will give rise to whole blood pool, all of the cells that are present in the blood cells. So let's now learn about the different cell lineages that can be formed by the hematopoietic stem cells. So now we are going to learn about hematopoiesis, means the generation of blood cells from the hematopoietic stem cells is known as hematopoiesis. When the hematopoietic stem cells they get differentiated and matured into blood cells, that particular process is known as hematopoiesis. So we are going to take a detailed look at the hematopoiesis. So hematopoietic stem cells, depending upon which type of transcription regulator is getting activated, they can commit to two cell lineages. I already told you that those two cell lineages are the myeloid lineage or the lymphoid lineage. So if the hematopoietic stem cell commits itself to the myeloid lineage, so it is going to form common myeloid progenitor. And if the hematopoietic stem cell commits to the lymphoid lineage, so that then it is going to form common lymphoid progenitor. Okay, so this myeloid, so this is CMP and this is CLP. So the CMP is going to give rise to erythroid progenitor and the myeloid progenitor, where the erythroid progenitor is going to give rise to RBCs. So it is going to give rise to erythroid progenitor, which will give rise to RBCs, the red blood cells, which are also referred as erythrocytes. Or it can commit, so this CMP, common myeloid progenitor, can commit to myeloid lineage by forming myeloid progenitor, right? So this myeloid progenitor cell can either it can form, it can develop and differentiate into multiple progenitor cells, which are going to commit themselves to being the WBC, to being the granulocytes in future. So the different progenitor cells that can be formed by the myeloid progenitor due to the differentiation are the granulocyte or granulocyte monocyte progenitor. Okay, they can form eosinophil progenitor, or they can form basophil progenitor cells, or they can form megakaryocytes. Right? The granulocyte monocyte progenitor cell, it can either form the granulocyte neutrophil or it can form the monocyte. Well, this monocyte can further in future form macrophage, or it can even get differentiated into dendritic cell. Okay, the eosinophil progenitor, obviously it is going to form eosinophils. The basophil progenitor, they are going to form basophils in future, while the megakaryocytes, if they get differentiated, they are going to form the platelets of the blood. Okay, so these are the different myeloid cells, the cells belonging to myeloid lineage that can be formed by the differentiation of the myeloid progenitor. On the other hand, the common lymphoid progenitor, they form the agranulocytes, the WBCs, which do not have granular structures. So they can be also differentiated into either dendritic cells, okay, dendritic cells, or they can form a B cell progenitor, they can form a T cell progenitor, or they can form natural killer cells. Okay, so the B cell progenitor can form B cells in future when they get maturated, they get matured, and when T cell progenitor they get differentiated, they can either form T helper cell or they can form cytotoxic T cells. Okay, so we will be discussing about those cells in detail in future lectures. So the only cell which belongs to both myeloid lineage and lymphoid lineage are the dendritic cells, which are also known as antigen presenting cells, and we'll have a separate lecture about those cells because they are really important and they belong to both myeloid lineage as well as lymphoid lineage. The myeloid cells, the cells belonging to myeloid lineages, means those granulocytes, those granulocytes, and the natural killer cells. Both of them, they belong to the innate immune system. On the contrary, the lymphocytes, such as B lymphocytes and T lymphocytes, they belong to the adaptive immunity. So the B cell mediated antibody production is going to result into the adaptive immunity. The cytotoxic T cell killing and the memory generation by the T cell that are going to result into the adaptive immune defenses. On the contrary, the myeloid cells, okay, so the myeloid cells as well as natural killer cells, both of them, they belong to innate immunity. So the cell mediated immunity, which is innate, it is established due to myeloid cells and the natural killer cells belong to the lymphoid lineage, and the cell mediated adaptive immunity, it is devised by the lymphocytes. Okay, so I hope this whole chart of hematopoiesis is clear to you all. So this is how hematopoietic stem cells, they can give rise to all of the blood cell types. So the isolation of those blood cells from the blood, it is a really unique process. So for the isolation of those different types of cells from the blood, we use a chemical reagent known as histopath. Okay, so histopath 1077, it is known as it is written 1077 because it has a density of around 1.0007 gram per ml. Okay, so this histopath, it enables the density dependent separation of blood cell. So what we do when we add the histopath in the blood cells, so what happens is that, let's assume that this is a test tube. Okay. Okay. So this is a test tube. Okay. And in this test tube, we have added blood. Okay. So the blood is diluted by some dilution factor. Let's assume that the blood has been diluted. So the blood and the histopath, both of them have different densities. So initially, they are going to separate into two phases. There will be blood separated from the histopath. Okay. And histopath is more dense. So this is blood. Okay, and this will be histopath initially. But once we centrifuge this whole solution is going to form different phases. So the very first, the lightest phase with least density will be the plasma. So it will be a clear solution, okay, which will be a little bit fuzzy, okay, a little bit fuzzy solution, translucent kind of, that will be the blood plasma. And then there will be a single layer of cells between the blood plasma and the histopath layer, which is below it. So this will be a clear solution, a clear phase, which is actually the histopath. Okay, and below that will be red blood cells. Okay, so below those, it will be a red solution, which is actually the red blood cells. And in this red blood cell solution, there will be some, so this will be red blood cells. Okay, so this solution will be reddish in color, and there will be some lighter colored layer over the red blood cells, which is also known as polymorphonuclear cells. So cells which are going to have multiple nucleus, they are accumulated in this particular region, in this particular phase. So in this phase belongs the agranulocytes such as neutrophils, basophils, eosinophils. So all of them, they get accumulated in this region, and the cells which are there, which are between the plasma and the clear solution histopath, they are referred as PBMCs. Okay, so those PBMCs are the cells which has, this is the pool of the cells which has the lymphocytes. So the major constituents of PBMC are the lymphocytes. So in this PBMCs belongs the agranulocytes. So PBMC stands for peripheral blood mononuclear cells. Okay, because they have single nucleus. Okay, so PBMC stands for peripheral blood mononuclear cells, and in these PBMC, major means almost 70% of the constitution of those PBMCs are lymphocytes. It also has monocytes, okay, means both of them they are agranulocytes, and they also have a few number of dendritic cells. Dendritic cells, they are present in a really less number in the blood. So a few dendritic cells, they are also a part of PBMCs. So we can say that the PBMCs has almost all of the different types of immune cells. So that is why in the immunological experiments, even in the immunotherapy experiments, we extract those PBMCs. So, PBMCs, they are separated from the blood and they are used for multiple different types of immunological experiments. So because they have lymphocytes which are going to have the, so we can separate the B cells, T cells from the portion, right? So the major fraction is the 70% of the PBMCs, they have the lymphocytes in them, while the layer between the histopac and RBCs is known as polymorphonuclear cells, and those cells has the granulocytes. Okay, so granulocytes are there in the polymorphonuclear cells, so such as neutrophils, basophils, and eosinophils. So they will be separate. We can also separate them out using the this whole method. So by this I'm done with the hematopoiesis as well as the hematopoietic stem cells. Okay, so let's explain one more topic. I would like to discuss about the isolation of hematopoietic stem cells. I already told you that the hematopoietic stem cells in bone marrow, they are very rare. So we can find one hematopoietic stem cell in each 50,000 bone marrow cells. So the isolation of the hematopoietic stem cells was really a critical challenge for the researchers, because back in the day there were not there were not that technical advancements. So once the flow cytometer got discovered, and then researchers got their hands on the formation of monoclonal antibodies, they could then isolate the hematopoietic stem cells. So for the isolation of hematopoietic stem cells, we use the elimination strategy, because so in a really great pool of the blood cell, in the really great pool of bone marrow cells, only a few cells, let's say one or two cells, are present in whole pool of the bone marrow cells. That is why the elimination strategy is best to isolate those cells. So these are the hematopoietic stem cells in this bone marrow pool, or in those bone marrow cells. So so it's like when you don't know the right answer, so you have been given a question and it has four options in the MCQ questions. What you do when you don't know the right answer, you go by the elimination strategy. Means you for sure know that those answers are wrong, so you are going to cross those answer, and eventually the answer you are left with is the right answer. In the same way, we use the elimination strategy for isolating the mature cells from the hematopoietic stem cells, because all of those other bone marrow cells, they are mature cells, while those hematopoietic stem cells, they are not differentiated, means they are undifferentiated cells, so stem cells. So scientists started looking for the markers that are present on the mature cells and the markers that are not there on the stem cell. So the markers, those are present on the mature cells, are known as lineage markers, okay, which is also abbreviated as LIN. So that is why mature cells, they were LIN positive, means they had this marker. And the hematopoietic stem cells, which are undifferentiated cells, they did not have those lineage markers because they are not committed to any type of lineage, right? Because they can be differentiated into any type of cell type, any type of cell lineage, and that is why the Lin negative markers were the hematopoietic stem cells. So this is how the elimination strategy was used to isolate the hematopoietic stem cells from the pool of the bone marrow cells. So the detailed method that they used is also known as panning. Okay, So this is the method of the isolation of the hematopoietic stem cells from this pool. So what did they do? They used the antibody which is bound to the plastic. So the lineage specific antibodies, and that is why the isolation of hematopoietic stem cell, it was possible only after the discovery of how to form monoclonal antibodies. Once we found the monoclonal antibodies, which with the anti lineage specific antibodies, so which can bind to specific lineage of the cells. So those antibodies, they are bound to plastic. So let's say that this is a tray, okay. So and in this is the plastic layer, and in here these antibodies are the plastic bound antibodies. All of them, they are specific to certain cell lineages, okay. Okay, and in this tray, I pour all of the cells that are there, all of the blood cells. I'm pouring all of the blood cells or the bone marrow cells in this particular tray. So those cells, most of the cells will be the mature cells which are belonging to specific lineages, and that is why all of the mature cells, they are going to bind to the antibody, okay. And a few cells will not bind to the antibody. Those few cells, okay, those only a few cells will be there, okay. Those cells, they do not bind to any antibody. They could be either progenitor cells, because progenitor cells, they are also the cells which are, they are also the cells which are not differentiated fully, so they are not mature yet, and that is why they are not going to have those mature cell markers, the lineage markers. But they are not stem cells, right? They are not hematopoietic stem cells. So both progenitor cells as well as the hematopoietic stem cells, both of them, they will not bind to those antibodies. So we discard the molecules which are bound to antibody, and we separate we give the wash, and we separate the unbound cells, okay. We discard those antibodies, so we discard the bound cells will be discarded because all of them are the mature cells. So we discard bound cells and we separate the unbound cells. So in those unbound cells, there will be hematopoietic stem cells as well as the progenitor cells. So when we have to differentiate between hematopoietic stem cells and progenitor cells, so researchers did a lot of study, a lot of research, and they found that the hematopoietic stem cells express certain markers, and the markers which are expressed by the hematopoietic stem cells are CD34, SCA1, okay, as well as c-KIT. Okay, so these are the different markers, these are the different surface markers which are expressed by the hematopoietic stem cells. So certain cell, if I'm saying that certain cell is CD34 positive, means it is expressing CD34. It is also expressing SCA1, or it is expressing c-KIT. This means that that particular cell is the hematopoietic stem cells. And that is why in nowadays, for isolation of hematopoietic stem cell, we use LSK phenotype. Okay, so LSK phenotype basically refers to the phenotype of the hematopoietic stem cell. Means if the cell has LSK phenotype, if a certain cell, that is 100% a hematopoietic stem cell. So what is this LSK phenotype? LSK phenotype is the cells which are Lin negative, okay, SCA1 positive, and K means c-KIT positive. If a cell has all those three characteristics, means it has Lin negative, so it does not express the Lin markers. It is Ka1 positive, means it expresses this marker, and it is c-KIT positive, means it also expresses c-KIT. So if a cell has those three characteristics, means it has a LSK phenotype, means that cell is the hematopoietic stem cells. So nowadays, for isolating the hematopoietic stem cells, researchers look for the LSK phenotype. Okay, and so when a mice, okay, so let's assume that this particular mice, okay, that this mice, this has been exposed to X-rays of about 95 rats. So this is going to kill all of the hematopoietic stem cells and the blood and the cells that are in the mice. So this mice will eventually die. So for saving this mice, for rescuing this mice from this fatal condition, we can either infuse it with the normal bone marrow cells. Okay, we can just introduce bone marrow cells in this mice, or we can even introduce those LSK phenotype cells means the hematopoietic stem cells, or we can also introduce the Lin negative cells. Okay, means the cells which does not have this Lin markers, means it could be either progenitor cells or it could be hematopoietic stem cells. So researchers, they carried out a study for what will be the survival rate of that particular mice if we infuse them, if we introduce either of those cells. So they plotted the observation of studies was something like this. So they plotted the survival rate, the percentage survival rate of the mice against the number of cells that are we are the number of cells that are we introducing in the mice. So let's say that this is 10, so it means that I am introducing 10 cells. On the x-axis, this is the number of cells that are being introduced in the mouse. So 10 raised to 1 means 10 cells, 100 cells, 1,000 of cells, 10 raised to 4 cells, or 10 raised to 5 cells. So it was already, so from the previous studies, it was already clear that when we infuse 10 raised to 4 to 10 raised to 5 bone marrow cells, the mice is going to survive. So if let's say, so the graph was something like this. So when we infuse so I'm going to show LSK phenotype with this color, and I'm going to show Lin phenotype, Lin mind negative cells with this color, and the bone marrow cells with this color. Means when the bone marrow cells, they were introduced in the cell. Let's say I'm introducing 10 bone marrow cells. The mouse did not survive. When I introduced 10 raised to 2, hundreds of bone marrow cells, again the mouse did not survive. In some cases it could, and in some cases it wasn't. So the survival of the mice started from almost 10 raised to 4 cells, and it increased as I started increasing the number of cells. And when the researchers, they introduced 10 raised to 5 bone marrow cell, it was 100%, it ensured the 100% guarantee of the mouse survival. Means if I introduce 10 raised to 5 bone marrow cell, it is for sure that mice is going to survive. Okay. And when I introduced the mature cell, when researchers started introducing the mature cells in the mouse, it the number of cells, the requirement of the number of cells, it decreased. So even if the mouse started surviving after, even after the infusion of hundreds of the mature cells. So even if I introduce 100 of the mature cell, the mouse started surviving. And as soon as I increase the number of cells, the percentage survival rate of my mouse increases. And once the thousands of this cells, they are introduced inside the mice, so now the mouse will have 100% survival rate. So this was the graph for the introducing the Lin negative, means the mature cells in the mice. So if the mature cells are being introduced, so those are progenitor cells and hematopoietic stem cells. So either of them are introduced. So number of cells that are going to be introduced in the mice, that decreases for ensuring the 100% survival of the mice. But when the LSK phenotype, means the direct introduction of hematopoietic stem cells was observed, the researchers observed that even if they introduce less than 10 hematopoietic stem cell, the mouse survived. Okay, so the survival graph started from here, and the even less than 10 or less than 100%, so only a few hematopoietic stem cells are required for ensuring the 100% survival of the mouse. So this was the graph which was found by researchers when they experimented the introduction of different cells. This is because the hematopoietic stem cells, they have enormous proliferation. So even a few cells, like 10 to 12 cells, 5 to 6 cells, they ensure the survival of the mouse. but when researcher introduces the lean negative cells, means they are introducing the somewhat mature cells. Okay, means somewhat differentiated cells. So they could be either progenitor cells or they could be either hematopoietic stem cells, right? We cannot really pinpoint whether they are progenitor cells or whether they are hematopoietic stem cells, but they are not the mature cells, and that is why it, yeah, number of cells that needs to be introduced for ensuring the 100% survival of the mice, that increased, right? So the number increased. But when the bone marrow cells, which are fully mature cells, so we need a lot of cells. In a lot of cells, there are the chances of finding one hematopoietic stem cells. So that is why the 10 raised to 4 to 10 raised to 5, this number of cells needs to be introduced in the mouse to ensure its survival, right? So as we introduce more and more mature cells, so these are the least matured, means totally undifferentiated cell. Those are somewhat differentiated cells, and these are fully differentiated cells. So as the differentiation of the cell increases, the number of cells that are required to ensure the survival of the mouse, that also increases, right? So with this, I'm done with the hematopoietic stem cells and the hematopoiesis. So in next lecture, we will learn about those particular stem cells such as neutrophils, eosinophils, basophils, the monocytes which are going to form macrophages in future, the B cells, T cells. So we will learn about all of those cells one by one in detail in next lecture. Okay. So with this, I'm done. I conclude the lecture here. Till then, see you. Bye bye.